different people subjects. Today we're going to discuss those macrophages in the immune response. Are we still awake after learning all these lectures from this morning? If something is not clear, then you can talk me. I will try to explain it a little bit further. So, macrophages, the name comes from the Greek. It means big eaters. Macrophages, macros mean large and phage meaning eat. And for a long time, it was thought that these macrophages did not much more than that. So they were thought of quite stupid cells, eating only maybe cell debris and pathogens. But now it has become clear that this is not true. These macrophages also exist like T cells in different phenotypes and have different characteristics and regulating the immune response in different ways. So today I'm going to very briefly discuss the discovery, the different subtypes that exist, the differentiation and the function. So I will explain just in theory the different receptors that are present on macrophages and the mediators that they can secrete. That may be a little bit more boring, but it's important to remember what kind of receptors these macrophages have to understand how they can react to different pathogens. So we'll discuss the activation and the phenotypes that exist, and in the end I will give you a few examples about the function of macrophages in wound healing, tumors, infection, atherosclerosis, and multiple sclerosis. So the macrophages were first discovered by Leonid Mechnikov, and he found out that the macrophages looked much alike the cells that were present in invertebrates that phagocytose took up particles and to defend these invertebrates against foreign substances. And at that time, it was still thought that the ingestion of for example, bacteria by macrophages was not to remove the bacteria, but led to the spread of the bacteria throughout the body. So there was quite some discussion about this at that time. But now it's clear that, of course, macrophages do this to remove these pathogens. And then there was Xanthor Cohn who discovered that macrophages and dendritic cells can take up antigens and present these antigens to T cells. And Charles Janeway discovered that these macrophages, also other cells of the innate immune system, possess pattern recognition receptors to recognize these pathogens and present them to T cells. And Simon Gordon, who's still a big name in the macrophage field, discovered that there can be different types of macrophage activation. That was only discovered about 5 to 10 years ago. So just to refresh your memory, macrophages belong to the innate immune system, like many other cells, and macrophages and dendritic cells look a lot alike. So sometimes it's not clear whether a cell is a macrophage or whether it's a dendritic cell. In general, it is said that when a cell can phagocytose a lot, then it's a macrophage, and when the main function is to present antigen, then it's a dendritic cell. And in many cases, this is not clear. So, compared to the adaptive immune system, macrophages and other cells of the innate immune system have only a limited amount of receptors that can, one receptor can recognize different pathogens. This is in contrast, of course, to the innate immune system, where each cell, each T cell, each B cell only has one receptor that can recognize one antigen from one type of microbe. The macrophages and the other cells have only about 100 different types of receptors, while in the innate immune system, of course, there are a million type of receptors. And every cell, same lineage, has similar receptors. Here it says identical, but of course the level of expression and some receptors are only present on certain cells of the immune system. But in general, the identical receptors are present, while in the innate immune system, there's only one receptor on one target receptor on B cell or B cell. But both have the ability to discriminate between cell and non-cell. So macrophages are derived from a stem cell, a hematopoietic stem cell in the bone marrow. So you don't have to remember all these names, but you have to remember that macrophages develop in the bone marrow, and after that they will enter the bloodstream as monocytes, and then can enter different tissues like the liver or the spleen, and then differentiate into different macrophage subsets. And for a long time it was thought that this figure is only from a few years ago that microglia cells also developed from this bone marrow stem cell, but now it's clear that this is not true. So I will explain this a little bit more in the end. So here these different macrophage subsets are shown. You have the Kupffer cells, for example, in the liver. In the spleen you have different cells, different macrophage subsets like the eosinophilic macrophages, and you have the osteoclasts in the bone that resorb bone tissue, and in the brain you have these microglia. So all these different subtypes are present and all have more or less different functions. They all can phagocytose cell debris and are important for when there's an infection to clear the pathogens, but they also have different specialized functions. Here you can see a few pictures of different macrophages. Here the alveolar macrophages in the lung, the osteoclasts in the bone marrow, and you can already see that they look quite different, but all here have these extensions to scan their environment for pathogens or for tissue debris. And here the most important functions of macrophages are shown. So they play a very important role in wound healing and the clearance of apoptotic cells. Sometimes this is not mentioned, but this is at least as important as the clearance of pathogens. So, for example, during T cell development in the thymus, of course, autoreactive T cells have to be removed. And also at the end of an immune response, when there are a lot of immune cells generated to clear pathogens, these immune cells have to be removed again when the pathogen is cleared. And of course, then it is important that this happens in an efficient way, and there the macrophages are very important. And also when you have, for example, a wound, this can happen without an infection, and there the cell debris has to be removed without generating a strong pathogen response. So of course the macrophages are also very important for pathogen removal. They can do this by phagocytosis, by the secretion of a whole range of mediators, and they can present antigen to activate the adaptive immune system. So I will discuss these mechanisms of pathogen removal very briefly. So when you look at phagocytosis, macrophages can take up particles in different ways. They can do so by endocytosis, by receptor-mediated endocytosis, and this means that proteins or small particles can bind to receptors on the surface of the macrophages and then will be internalized. Once internalized, they will fuse with endosomes, and in the end they will fuse with lysosomes where the proteins are degraded and materials are resolved. These proteins will also be presented on the cell surface by MC2 molecules to present them to the helper cells. They can also macrophages can also take up particles by phagocytosis, meaning that very small molecules will be engulfed by the membrane of the macrophage and then taken up in this way. And they can phagocytose larger particles also by engulfing them, and this can either be receptor-mediated or non-receptor. In case of apoptotic cells, when they receive an apoptotic signal, they will present phosphatidylserine on the surface. And this is a signal for macrophages that these cells have to be removed. So this phosphatidylserine will bind to receptors on the surface of macrophages, and then cell will be taken up and will be degraded into smaller particles. And these particles, of course, can be very useful to be used for other processes. And also, it is important that in case of apoptosis, that not an inflammatory response is generated, and therefore the macrophage will secrete anti-inflammatory cytokines like IL-10 or TGF-beta, and will downregulate the expression of MC2. So no T cells will be activated in this case. Macrophages secrete a plethora of mediators like oxygen radicals, complement, cytokines, but also other factors like chemokines, neurotransmitters, and lipid metabolites, for example. I will only discuss the first three now. So when macrophages start phagocytosing bacteria or cell debris, the NADPH oxidase complex will be activated. This will lead to the production of oxygen radicals like superoxide or hydrogen peroxide. And these oxygen radicals are very important, first to damage the bacteria, for example, to the bacteria, but they are also very important to stimulate the phagocytosis process, so that you take away these oxygen radicals, no phagocytosis will occur, and they will activate signaling cascades to activate the macrophage. So these radicals are very toxic, and the cell also possesses some enzymes to protect the cell against these oxygen radicals, like catalase. Yeah. So the macrophage also produce complements, and I think most of you already know what complement does. It's something from your bachelor's, but just to refresh your memory, you can see here that complement can be activated in three different ways. For example, by the lectin pathway, by antibodies, this is the classical pathway, and by spontaneous activation on surfaces that do not possess complement inhibitors. So this will lead to the activation of complement factor C3, and this is amplified, this reaction, and then different effective mechanisms can occur, like the secretion of anaphylaxis toxins, which lead to chemotaxis of different immune cell subsets. The membrane attack complex can be formed to damage bacteria, for example, and the complement can bind to antibodies and bind to complement receptor on the macrophage that lead to phagocytosis and clearance of infections. Is this clear? So macrophages also produce cytokines. I just shown a few examples like TNF alpha, IL-1 beta, IL-12, IL-6. These are all pro-inflammatory cytokines that play an important role in regulating the immune response. For example, IL-12 uses Th1 type of immune response, stimulates T cells and NK cells. While TNF alpha is well known for activating macrophages, but also for inducing apoptosis. Then you have anti-inflammatory cytokines, like IL-4 and IL-10. These cytokines induce a Th2 response, and can also be neuroprotective and can stimulate PKS. So when macrophages phagocytose pathogens, it is important that these molecules derived from these pathogens are presented to T cells to elicit the adaptive immune response. So once these pathogens have been taken up and have been degraded inside of the macrophage, they are presented by MC class 2, And this, together with the presentation of the co-stimulatory molecules, specific T cell subsets can be activated. And that's something about the receptors present on the membrane surface of macrophages. So you have different groups of receptors. You have receptors that are directly involved in phagocytosis, like complement receptors, Fc receptors, lectins, scavenger receptors. And you have the toll-like receptors that are very important in sensing the environment and directing the macrophage response and the phenotype in which the macrophages will develop. So when we look at the phagocytosing receptors, you have receptors that can directly recognize pathogens, like the pattern recognition receptors, and like scavenger receptors and lectins. So these receptors can bind directly to certain groups of molecules on the surface of, for example, bacteria, and then these receptors will internalize together with the bacterium and then degrade the pathogen. But you have also receptors that can indirectly lead to phagocytosis, like the complement receptors and the Fc receptors. So I will explain this a little bit further. Concerning the scavenger receptors, you have a whole range of different types of scavenger receptors. You don't have to remember these. It is important to remember that they can directly bind to bacteria and can induce phagocytosis and cytokine expression. This also counts for lectins. Lectins bind to sugar groups. And these sugar groups, binding to the sugar groups can lead to phagocytosis, but in contrast to scavenger receptors, lectins have a whole range of other functions, like they are important for adhesion to endothelial cells. For example, when macrophages migrate through these endothelial cells to sites of infection, they are important for cytokine production, also for antigen presentation, and you have also lectins that can be secreted in case of these collectins, and then can bind to bacteria and induce phagocytosis. Concerning the Fc receptors, so these receptors are important for antibody binding, and when an antibody has bound to a pathogen, these antibodies will cross-link, bind to a Fc receptor and cross-link them, and then will give a signal inside the macrophage. And depending on the type of receptor that is activated, you can either have an activation signal or an inhibitory signal. So here, this is again an example that shows that it matters a lot which kind of receptor is activated, and depending on the range, the different receptors that are activated on a macrophage, a certain type of response will be generated. And this also comes for the complement receptors. So you have, again, a whole range of complement receptors, and depending on the receptor that is bound, phagocytosis can be induced, macrophages can migrate in case of the anaphylatoxin receptors, and they can modulate cytokine production. And finally, something about the toll-like receptors. And toll-like receptors do not induce phagocytosis, but direct the macrophage response into a certain direction. The most known toll-like receptors are, for example, TLR4 that binds to lipopolysaccharide, and the TNF receptor that can induce phagocytosis. But these receptors can bind to foreign particles, but it's now also shown that these receptors can bind to endogenous ligands, and then, again, depending on the type of receptor that are activated, either an activating signal is generated or an inhibitory signal. So, as I told you, macrophages can be activated in different ways, generating different phenotypes. And the most recent years, there has been a lot of discussion on what kind of macrophage subsets, what kind of macrophage phenotype do exist, and what their function is. So it has been suggested that there are M1 and M2 macrophages, being two extremes on a whole range of subtypes that exist in between. But it could also very well be that there are more subtypes present, and that, for example, classical activated macrophages, regulatory macrophages, and wound-healing macrophages, and a macrophage can be somewhere in between these types of phenotypes. So in contrast to T cells, where you have a clear subset of T1, T2, T17 cells, macrophages exist in a whole range of phenotypes, where there may be some extremes, but macrophages can also exist in between a subset, and can also it's also clear that macrophages can differentiate from one phenotype to another phenotype if it receives a different signal. This is also different for T cells. So the phenotype that is generated depends first of all on the macrophage subtype. So whether it's a macrophage that exists in the lung, for example, whether it's an avidiolar macrophage, whether it's a microglia or a monocyte-derived macrophage. But it also depends on the surrounding tissue, the signals that the macrophage receives from this tissue. It depends on the cytokines that are present in the environment, and it also, of course, depend on if they are present, on the pathogen that is recognized by a macrophage. But then, depending on the signals that these macrophages get, either as it is thought now, but this can change in the next few years maybe, that there exist classically activated macrophages, and these are also called M1 macrophages, and these are generated by interferon gamma or LPS, lipopolysaccharide. You have the wound-healing type of macrophages that are generated by IL-4, and you have a more regulatory type of macrophage that is thought to be generated by IL-10. So these are the most potent inducers of this phenotype, but by phenotypes, but of course, other cytokines and signals also play a role. Here these phenotypes are shown again, and here it is shown how these phenotypes can direct T cell responses. So the classically activated macrophage treats IL-12 that will generate Th1 cells, but can also produce IL-6 and IL-23 that are important cytokines for inducing Th17 cells. The regulatory macrophage that is induced by IL-10 can also produce IL-10, which will induce Th2 cells, and these Th2 cells will then produce IL-4 and IL-13 that will then induce the wound-healing type of macrophage. So this is something that is important to remember. And I will show you a few examples of the role of macrophages in different diseases and other types of repair, for example. Concerning infection, the type of macrophage that a response that is generated depends a lot on the type of infection. For example, when you look at viruses and bacteria, mainly M1 responses are activated. This is just an example, so you don't have to remember this picture completely. But for example, when you have a Leishmania virus that is inserted by the sandfly, then this virus will be taken up by macrophages. But this type of infection will downregulate the macrophage response. So this alone will not generate an effective pathogen clearance. But in the meanwhile, also complements will be bound to this virus, and then cytokines are produced that will attract monocytes from the blood and will lead to an effective macrophage activation and one activation that will lead to clearance of the pathogen. So, and in case of a warm infection, maybe an M2 response is generated, and this M2 response will be generated by the secretion of recruited Th2 cells that produce IL-4 and lead to the activation, differentiation of macrophages in M2 macrophages that will then induce killing of the wall. When the pathogen is not effectively cleared, then a granuloma can be formed. This means that the macrophages that are present will fuse and mature into multinucleate cells, and these cells produce cytokines that will attract other immune cell subsets like neutrophils, other macrophages, T cells, and these T cells will be activated by antigen presented by these macrophages, and this will lead to a granuloma in which the pathogen is encapsulated, and in this way the pathogen cannot spread further throughout the body, and hopefully in the end the pathogen will be cleared. Yeah, then something about wound healing. This scheme already shows what can happen when the skin, for example, is damaged, and then of course pathogens are allowed to enter the body, but this can also happen without the presence of pathogens, of course. But initially, macrophages present in the skin, dendritic cells present in the skin, will take up the pathogens, try to clear them, and will in the meanwhile secrete cytokines, chemokines that will attract other cells from the blood to the site of infection to clear the pathogen and to induce wound healing. This is a scheme what happens during this wound healing response. So you have here the epithelial cells that are damaged, and then two types of responses are induced. First of all, the dendritic cells and macrophages present will secrete IL-12, and this will induce an M1 type of macrophage, leading to pathogen clearance and the activation of T cells. But when this, in the meanwhile, also another response is generated by the secretion of IL-25, IL-33, and this will activate mast cells, Th2 cells, and basophils that will produce IL-4 and IL-13, generating an M2 type of response. But as long as this response is present, and as long as the pathogen is present, this response will suppress the M2 type of response. So the first goal is to remove the pathogen, and once the pathogen is cleared, then this response will fade out, and this response will dominate, and that will then induce certain growth factors that can induce repair of the damaged tissue. So this is an overview of what happens. So there you have, during wound healing, you have the basophils and Th2 cells that produce IL-4 that induces wound healing by the production of extracellular matrix proteins. Also then, no antigen will be presented anymore, as the pathogen is already effectively removed, and you have a very low production of pro-inflammatory cytokines and repair-inducing cells. In case of a tumor formation, the tumor actually uses the fact that macrophages can differentiate into wound healing macrophages, because the tumor, this resembles a tumor, induces factors that attract macrophages from the blood and differentiates these macrophages in more M2 type of macrophages. And in this case, they are called tumor-associated macrophages. And these type of macrophages secrete factors that lead to angiogenesis and to factors that stimulate the tumor growth. Also, as they are more M2 type of macrophages, no effective antigen fragmentation occurs, and therefore the tumor is not removed very well, and therefore these macrophages play a detrimental role in the tumor formation. And therapies focus on how to change this macrophage phenotype into a more normal type of macrophage, and so that the tumor can be effectively be addressed and removed. Then something about atherosclerosis. So this is a very complex scheme, but it just shows what happens during atherosclerosis. So when you have high lipid levels in the blood, then these lipids will lead to a dysfunction of endothelial cells, and leukocytes will slow down and will enter, will leave the bloodstream and will enter the subvascular space. And in this space, these immune cells will take up lipids and will differentiate into foam cells, and these foam cells secrete all kinds of factors that attract other immune cells like T cells, and then a plaque is formed. And in the center of the plaque, you have necrotic tissue, and you have a fibrous cap that keeps the atherosclerotic plaque together. But in the end, this fibrous cap can rupture, can occur, and then of course it can be thrombosis, and people can die from this event. So what's the role of the macrophages in this process? Well, so you have the high LDL, the high lipid levels present in the blood, and this LDL can bind to different types of receptors. And especially when this LDL is oxidized, certain types of scavenger receptors are activated, and this will lead to activation of the inflammasome and secretion of pro-inflammatory cytokines and other molecules that will all together lead to stimulation of the immune response and to worsening of this atherosclerotic plaque. And finally, something about macrophages in the central nervous system. So you may all have heard about microglia and macrophages that can enter the central nervous system during infection. But actually there are more types of macrophages. You don't have to remember this all, but they reside at different sites in the central nervous system. For example, in the meninges, in the choroid plexus, and all have specialized roles. For example, these perivascular macrophages around blood vessels. Sometimes they are called dendritic cells, sometimes they are called macrophages. But these cells play an important role in reactivating T cells that enter the central nervous system during an infection. There has been a lot of research and discussion about where these microglia come from. So it was not clear whether these microglia are replaced by monocytes from the blood, and then these monocytes could in time have to enter the central nervous system and could in time differentiate into microglia. And this was thought to be the case, and to prove this, quite complex experiments were performed. For example, mice that were positive for GFD were used, and bone marrow was transferred into non-fluorescent mice. This was done by, of course, isolating the bone marrow that contains the green fluorescent macrophages, and transplant this bone marrow in mice that were irradiated. And this irradiation leads to the depletion of all the macrophages in the system of the mice, but does not affect the microglia in the central nervous system. So when this bone marrow was transferred to these mice, it was thought that people could check whether these macrophages from these green fluorescent macrophages would in time enter the central nervous system and replace microglia. So if fluorescent cells would be present in the central nervous system after a few months, then it could be concluded that these microglia were derived from monocytes, and not from another source. And indeed, these cells were found, so that was what was concluded. But now it has been shown that this is not true. What actually happened is that, first of all, during the evaluation, the blood-brain barrier is damaged, and the bone marrow that was used did, of course, also contain a whole range of different stem cells, and the stem cells were able, when the bone marrow was injected in the bloodstream of these mice, were able, due to the blood-brain barrier damage, were allowed to enter the central nervous system. So this was actually a non-physiological thing that happened using this method. Because what was recently done is that even a more ingenious method was used. And for this, two mice were sewed together. The blood vessels of these mice were sewed together. And one mouse was the green fluorescent mouse, while the other mouse was just a wild-type mouse. And after a few weeks, one mouse was irradiated. So in this mouse, all peripheral monocytes were depleted. And then, after a few weeks, these depleted cells were replaced by the cells of the green fluorescent mouse. But now these cells were directly derived from the bloodstream and not from the bone marrow, where the stem cells are present. Yeah, is that clear? So now we have a mouse, and these are separated again, where the peripheral cells are green fluorescent that are not derived from stem cells. And in this case, no green cells were detected in the central nervous system. So then it was clear that the microglia that are present in the central nervous system are not derived and are not replaced by blood monocytes. And so where do these microglia come from? So these microglia are derived from the yolk sac progenitors and already enter the embryo during development. And then once in the central nervous system, they are not replaced by monocytes, but divide inside the central nervous system once there's an infection, for example, and very slowly divide in time. And at the end of the immune response, these microglia will die. And during an infection, these microglia will divide, and also monocytes from the bloodstream that will mature in the bone marrow will enter the central nervous system and will help to clear the infection. So in the central nervous system, you have two different types of macrophages with a different origin that help to clear infections. Then I'm going to tell you something about MS. I think most of you will have heard this maybe a million times, but I will very briefly give an introduction to MS. So MS is a chronic inflammatory disease of the central nervous system with a 2:1 female to male ratio. And up to now we don't know the cause of MS, but probably it is a combination of both genetic and environmental factors. During MS, an autoimmune response is directed against myelin components that causes demyelination and failure of external production. And we have no cure available yet, and can only help these patients by symptomatic treatment. So what happens? You have autoreactive T cells that are activated due to an unknown cause. They will enter the central nervous system. They'll be reactivated by antigen-presenting cells over there, and will secrete chemokines that will attract monocytes from the blood. And these monocytes will differentiate into macrophages and demyelinate axons. And this then leads to clinical symptoms. And during MS, we can detect these macrophages and T cells that have entered the central nervous system by MRI, which helps to diagnose NS. And when we look at these sites of infection, we see that these are initially filled with macrophages. And these macrophages, these macrophages seem to play an important role in the initiation and progression of MS, but how can we study this? So for this we can use different systems. We can make use of, for example, lumen material, but when we want to have brain tissue from an MS patient, we have to wait, because MS is not lethal, till these patients die. And then these patients will already have MS for about 20, maybe 40 years, and are in an end stage of the disease. So this tissue does not allow us to understand what happens in the initial stage of the disease. But of course, this tissue helps a lot to understand the mechanisms that are going on. But what we can do is that we can isolate monocytes and also T cells, of course, from the blood of MS patients and study whether these cells differ from cells in healthy persons. But we can also make use of in vitro models. We can make use of cell lines, tumor cell lines of macrophages, T cells, to study how they react to certain triggers. We can make use of primary cells. These cells can be, for example, macrophages, T cells, isolated from mice, and also oligodendrocytes, neurons isolated from mice. But we can also, of course, use these monocytes from MS patients and see, differentiate them into macrophages and see how they react to certain triggers. We can also make use of co-culture models that are more complex, for example, models of oligodendrocyte neurons, and co-culture them with macrophages to see what happens. And of course, we can also make use of animal models for MS to study if certain new therapies are effective, and to study the mechanisms that are going on in a living creature. So when we look at the monocytes isolated from MS patients, we can already see that these monocytes are more activated. Here you see MS patients, here control patients, and shown are the nitric oxide levels. And then it's clear that macrophages, monocytes, I have to say, isolated from the blood from MS patients already produce more nitric oxide. And this also accounts for other inflammatory cytokines like IL-1, TNF-alpha from gamma, and these are produced at higher levels, while anti-inflammatory cytokines like IL-10 and TGF-beta are produced at lower levels. So this already proves that there is a difference between monocytes isolated from MS patients compared to those isolated from healthy controls. We then also see that when we look at electron microscopy, that monocytes migrate from the bloodstream to the central nervous system. This was of course what is expected when we see that macrophages are present there. And we see that at the sites where these macrophages are in the central nervous system, that myelin disappears. So this is, in blue you see the myelin. There's a big lesion, a big MS lesion, where all the myelin has been removed. When we look more closely at these macrophages and stain for lipids, we see that these macrophages at the rim of an MS lesion are filled with myelin. So then it seems clear that these macrophages are cells that are doing damage inside the central nervous system by removing this myelin. But it's not that easy to conclude. What's also clear is that at the site where macrophages have infiltrated, also neurons and axons are damaged. So these macrophages, once enter the central nervous system, take up myelin, remove myelin, and produce all kinds of inflammatory mediators that can induce demyelination and axonal damage. And so for the last maybe 100 years, we saw that these macrophages were only detrimental in MS. So they produce bad toxic factors, they affect the myelin, but more recent research has shown that this is not completely true, because when we look at macrophages in different sides of an MS lesion, so this is the center of the lesion, this is the rim of the lesion, this is normal CNS tissue, we see that these macrophages, especially in the center of an MS lesion and at around perivascular spaces, also produce a lot of anti-inflammatory factors, and even factors that can induce repair. So not every macrophage in an MS lesion has the same phenotype and has the same role in the progression of the lesion. To study what the function is of macrophages in neuron inflammation and in demyelination, we have to use an animal model. And you may have already heard about the EAE model, which means experimental autoimmune encephalitis myelitis, that is induced by a subcutaneous injection of myelin protein and adjuvant. And this will then induce an immune response leading to different clinical symptoms depending on the myelin protein that is used and on the animal strain that is used. So you can have, there are models that in which only a short period of clinical symptoms is induced, and animals are paralyzed and then are recovered again. There are also models in which relapses occur, and models that have more of a chronic disease type. So what happens when we deplete macrophages from an EAE mouse or rat? This is something what we cannot do that easily, of course, in humans when we don't know what happens. When we deplete macrophages by using chloromnate liposomes, chloromnate is a toxic substance that, when taken up by macrophages, will lead to cell death. And these liposomes are specifically taken up by macrophages by phagocytosis, and so these macrophages are the only cells that are depleted. When we use these liposomes, we see that the clinical symptoms, as shown here, will almost completely disappear. So it's clear that the macrophages, at least initially, play a detrimental role in neural inflammation. However, when we inject, for example, myelin debris inside the central nervous system, we see that this myelin debris will prevent repair, prevent remyelination at the site where this myelin is injected. When macrophages are then depleted during this injection of myelin debris, we see that less remyelination occurs. So this means that removal of myelin debris is essential to induce repair of damaged sites. And in this case, macrophages do play a beneficial role. So to study this further, we can make use of in vitro model. We can, for example, add myelin, here shown in red, to macrophages in culture, and this myelin will be phagocytosed with macrophages, and we can then see what happens with the phenotype of these macrophages. We can then co-culture these macrophages with, for example, autoreactive T cells and with oligodendrocytes in culture. What you then see is that when myelin is taken up by macrophages, these macrophages start to produce less nitric oxide and also less IL-6. So here's the control level. And you can see that when this myelin is added, less inflammatory mediators are produced. And when we co-culture these macrophages together with autoreactive T cells, we see that also less T cell proliferation is inhibited. So here are control macrophages, here are myelin phagocytosis macrophages, and even when these macrophages have taken up myelin, the proliferation of T cells is even reduced further. So this can also be a beneficial effect of macrophages on the autoimmune response that is going on. This was just a pilot experiment that shows that oligodendrocytes are cultured and macrophages are added. These macrophages can induce oligodendrocyte damage. So this is the viability shown on the y-axis. But when these macrophages are co-cultured with myelin, they are less toxic to oligodendrocyte. So this altogether shows that when macrophages are take up myelin, they can at first be maybe detrimental for oligodendrocytes, for neurons, but after this myelin uptake, they will change their phenotype into a more wound-healing type of macrophages, and this can induce repair. So we were interested what kind of factor in this myelin is responsible for this change in macrophage phenotype. For this, we co-cultured the macrophages with myelins, but also with different lipids present in myelin. And then we found that certain lipids present in myelin could regulate the macrophage response. Here you can again see the effect of myelin on the secretion of pro-inflammatory mediators by macrophages, and you see that TNF-alpha is reduced, and also this is a marker for N2, a wound-healing type of macrophage, and this is a repair-inducing cytokine. You see that myelin suppresses inflammation, but that certain lipids present in myelin have even stronger N2-inducing phenotype and could lead to the secretion of repair-inducing cytokines. So it can be that myelin contains, of course, different lipids, different proteins that can have also a differential effect on the phenotype that is induced. And when we inject these lipids in the animal model, we also see that the macrophage response is not only altered in vitro but also in the animals, and that clinical symptoms of these animals, disease symptoms, are reduced when we inject this lipid present in myelin. So this was what I wanted to show you. What we can conclude from the last part is that macrophages are important effector cells. They can present antigen, they can secrete toxic factors, but they can also clear myelin debris and produce toxic factors and thereby modulate immune responses. There are different therapeutic options that address macrophages that are currently being studied to treat MS patients. First of all, there are treatments that are already present that block macrophage infiltration and thereby may prevent relapses. There are also therapies that can that inhibit macrophage activity, but as I showed you, there may also be beneficial macrophages present, and then we prevent their infiltration, prevent their activity in the central nervous system. This may be detrimental for demyelination and repair. So therefore, it might be more interesting to modulate the phenotype of macrophages, which can be done, for example, with lactic acetate. And therefore, we hypothesize that maybe during MS, the macrophages and macroglia are activated and produce pro-inflammatory cytokines and damaging factors. But it might be interesting to develop new therapies that will restore the macrophage phenotype into a more wound-healing type of macrophages, and thereby the balance that is disturbed during in MS patients in the central nervous system might be restored. So, I would like to end by giving you some take-home messages. The macrophages are very important to clear infections, to regulate immune responses, and induce repair of damaged tissue. They possess a whole range of mechanisms that are by which they are able to recognize and clear pathogens. These macrophages can occur in different subsets and different phenotypes, and these phenotypes can be changed and can be induced depending on the local microenvironment. And I hope I've showed you that these macrophages might be interesting targets to modulate the disease process and the inflammatory response. That's it. Are there any questions? Okay, then we can have a short break. And then I think in 15 minutes we will go on with the next lecture.